At the end of this topic, you will be able to point out the source of energy, define what respiration is, categorize the respiratory substrates, differentiate aerobic respiration from anaerobic respiration, discuss the steps involved in respiration, calculate the amount of ATP released during respiration. Sunlight is the source for our food in this planet. Food that we eat is the starch that is prepared by plants during photosynthesis. Food that we eat is the source of energy, and the energy is stored in our body in the form of ATP, which is adenosine triphosphate, which is commonly known as energy currency of the cell. We need energy for growth, movement, reproduction, and other activities. Respiration is the process of conversion of useful nutrients into useful energy. The energy is released from the food during respiration, and it is stored in a special biochemical substance called ATP or adenosine triphosphate. Carbon dioxide and water are the byproducts of respiration. C6H12O6, which is glucose, plus 6O2 gives 6CO2 plus 6H2O plus 2900 kilojoules energy, which is ATP. Substance that is used in respiration is known as respiratory substrate. There are three kinds of respiratory substrates, namely carbohydrates, fats, and proteins. Basically, there are two types of respiration, aerobic respiration and anaerobic respiration. Aerobic respiration is carried out in the presence of oxygen. Anaerobic respiration is carried out in the absence of oxygen. Aerobic respiration takes place in 4 stages: glycolysis, oxidative decarboxylation of pyruvic acid, Krebs cycle, electron transport chain. Glycolysis takes place in the cytoplasm of the cell. In glycolysis, glucose, a simple carbohydrate, is split into 2 molecules of pyruvic acid. Glycolysis is a series of reactions and number of enzymes involved in it. During glycolysis, 2 molecules of ATP and 2 molecules of NADH, nicotinamide adenine dinucleotide, hydrogenase, is released. 1 NADH releases 3 molecules of ATP, so the formation of ATP at the end of glycolysis is 8.
Glycolysis. Glycolysis literally means splitting sugars. In glycolysis, glucose, which is a 6-carbon sugar, is split into 2 molecules of a 3-carbon sugar. Glycolysis yields 2 molecules of ATP and 2 molecules of pyruvic acid. Glycolysis can occur with or without oxygen. In the presence of oxygen, glycolysis is the first stage of cellular respiration. Without oxygen, glycolysis allows cells to make small amounts of ATP. This process is called fermentation. Let's look at glycolysis in detail. In the first step of glycolysis, glucose is converted to glucose 6-phosphates in the cell's cytoplasm. The enzyme hexokinase phosphorylates and adds a phosphate group to the glucose molecule. During the reaction, a phosphate group from ATP is transferred to glucose producing glucose 6-phosphate. In step 2 of glycolysis, the glucose 6-phosphate is converted into its isomer fructose 6-phosphate. Phosphoglucoisomerase acts as the enzyme during this reaction. Isomers have the same molecular formula, but differ in the arrangement of atoms in the molecules. In step 3, the fructose 6-phosphate is then converted to fructose 1,6-diphosphate with the presence of the enzyme phosphofructokinase. 1 ATP molecule is used to transfer the phosphate group to fructose 6-phosphate. The 6-carbon compound fructose 1,6-diphosphate is broken down into 2 3-carbon compounds that are isomers of each other in step 4 of glycolysis. These 2 sugars are dihydroxyacetone phosphate and glyceraldehyde phosphate. In step 5 of glycolysis, the dihydroxyacetone phosphate is converted to its isomer glyceraldehyde phosphate. The enzyme triose phosphate isomerase catalyzes the reaction. In step 6 of glycolysis, the enzyme triose phosphate dehydrogenase transfers hydrogen from glyceraldehyde's phosphate to the oxidizing agent nicotinamide adenine dinucleotide to form NADH. In the same reaction, the enzyme adds a phosphate from the cytosol to the oxidized glyceraldehyde phosphate to form 1,3-diphosphoglyceric acid. These two functions occur for both the molecules of glyceraldehyde phosphate. In step 7 of glycolysis, an ATP molecule is formed by the transfer of a phosphate group from 1,3-diphosphoglyceric acid. One ATP molecule is converted to ATP. This happens to both the molecules, leading to the formation of two molecules of 3-phosphoglyceric acid and two ATP molecules. In step 8 of glycolysis, the enzyme phosphoglyceromutase relocates the phosphate from 3-phosphoglyceric acid from the third carbon to the second carbon to form 2-phosphoglyceric acid. In step 9 of glycolysis, 2-phosphoglyceric acid is converted to phosphanol pyruvic acid by the enzyme enolase. The enzyme removes a molecule of water from 2-phosphoglyceric acid. This happens to each molecule of 2-phosphoglyceric acid. In step 10, the enzyme pyruvate kinase transfers a phosphate from phosphoenol pyruvic acid to ADP to form pyruvic acid and ATP. This reaction yields two molecules of pyruvic acid and two ATP molecules. Thus, at the end of glycolysis, two molecules of pyruvic acid and two molecules of ATP are formed.
Oxidation of pyruvic acid takes place in mitochondria of the cell. Pyruvic acid formed at the end of glycolysis is oxidized to form acetyl CoA. During the pyruvic acid oxidation, 2-NADH, nicotinamide adenine dinucleotide hydrogenase, and carbon dioxide is released. Pyruvic acid oxidation is continuous with Krebs cycle which starts from acetyl CoA. There are events of cyclic reactions in Krebs cycle which takes place in the mitochondria. During Krebs cycle, 6 molecules of NADH, 2 molecules of FADH2, flavin adenine dinucleotide hydrogenase, and 2 molecules of ATP is released. 1 FADH2 in Krebs cycle releases 2 ATP molecules. At the end of the Krebs cycle, there is a formation of 24 ATP molecules from ATP, NADH and FADH2.
ATP synthesis. This animation shows the continuous cycle of adenosine triphosphate, which is known as ATP production. Glucose molecules generally float around in the cytosol until they undergo glycolysis to convert them to pyruvic acid. This pyruvic acid then enters the Krebs cycle. Chemical energy within the mitochondrion forces the products of the Krebs cycle into the electron transport chain. Here, it reacts again in order to produce a large amount of ATP. As seen here, ATP synthesis takes place in the mitochondria of a cell. The mitochondria consists of three parts, namely the inner membrane, the outer membrane, and the matrix. In mitochondria, the protons are translocated across the membrane from the matrix to the inner membrane space. This helps in the formation of NADH by oxidation reactions. Thus, the continued buildup or excess of these protons creates a proton gradient across the membrane. The re-entry of protons is allowed by the proton channel of a large protein complex called ATP synthase. Thus, protons enter the mitochondrial matrix through the channels in the ATP synthase enzyme complex. This entry of protons into the channel triggers the synthesis of ATP from ADP to adenosine diphosphate and inorganic phosphate, shortly called Pi. This synthesis is facilitated by the resulting current of protons flowing through the membrane. Thus, ADP and inorganic phosphate combine to produce ATP.
Electron transport chain is carried out in the inner membrane of mitochondria since it has all the enzymes which are needed for transporting electrons. During the electron transport chain, the energy associated with the liberated electrons is used to synthesize ATP at certain stages. Finally, the hydrogen and electron joins with oxygen to produce water as a byproduct. One NADH releases 3 ATP molecules and one FADH2 releases 2 ATP molecules. The net gain of complete oxidation of a glucose molecule in aerobic respiration produces 38 ATP molecules.
Electron transport system. Electron transport system involves the production of high-energy phosphates or the ATPs in different phases. In the first phase of the process, one molecule of ATP is produced from NADH. On the other hand, NADH2 is reduced by the substrate, which helps in the formation of ATP. In the second phase of electron transport system, the FADH2 molecule produced in the TCA cycle is converted to FAD. This helps in the formation of ATP from ADP. Flavoproteins and cytochrome B are involved as electron transporters in the cycle. In the third phase of the cycle, one ATP molecule is produced by the electron transported by cytochrome C and cytochrome A. In the fourth phase of the cycle, the protons produced in phase 2 react with the oxygen to form water.
Anaerobic respiration is otherwise called fermentation. There is no consumption of oxygen in respiration. Only glycolysis is carried out and other steps of respiration are completely off. The end product of glycolysis is ethanol or lactic acid. Only 2 ATP is released, so the energy released is quite low. A very common example for fermentation is conversion of milk into curd.
In this topic, you have learned
we get our food from plant and food is essential for growth and other activities. Respiration is the process of conversion of useful nutrients into energy which is released in the form of ATP. Carbon dioxide and water are the byproducts of respiration. The substance that is used for respiration is known as respiratory substrate. There are 2 types of respiration, namely aerobic respiration, which is carried out in the presence of oxygen, and anaerobic respiration, which occurs in the absence of oxygen. Aerobic respiration consists of 4 steps, namely glycolysis, oxidation of pyruvic acid, Krebs cycle, electron transport chain. Glycolysis takes place in the cytoplasm, in which glucose splits into 2 molecules of pyruvic acid and releases 8 molecules of ATP. Oxidation of pyruvic acid, Krebs cycle and electron transport chain carried out in mitochondria. 6 ATP is released during pyruvic acid oxidation and 24 ATP is released during Krebs cycle. ATP is synthesized by using electrons in electron transport chain and releases 38 ATP molecules. In anaerobic respiration, only glycolysis is carried out and releases carbon dioxide, ethanol or lactic acid and 2 molecules of ATP.